It is estimated that by 2050, antibiotic-resistant superbugs will be responsible for the death of 10 million people per annum. This clearly indicates that this is a huge problem that needs to be solved. In this video, we will explain how these bacteria acquire this resistance, but first we must discuss the different sources of antibiotics, their two main types, and their different mechanisms of action. Firstly, antibiotics are either synthetic chemicals or natural products. The majority of these antibiotics are natural products of bacteria and fungi due to their competition with other bacteria for space and nutrients. Secondly, antibiotics are classified as either bacteriostatic or bactericidal according to their function. Using a line graph of number of bacteria versus time, we can summarize their differences. The line represents the increasing number of bacteria over time due to reproduction or replication. After the addition of the drug, a change occurs. Bacteriostatic antibiotics inhibit bacterial growth, therefore the number of bacteria does not change over time, while bactericidal antibiotics inhibit their survival, basically killing them, therefore their numbers decrease over time. Thirdly, an antibiotic's mechanism of action exploits the differences between bacterial cells and human cells. Here we have a typical bacteria with an outer cell wall and a plasma membrane. Its DNA is located within the cytoplasm of the cell, and usually RNA is being synthesized off it. In addition, ribosomes are dispersed throughout the cytoplasm of the bacterium, and these are responsible for the synthesis of bacterium's proteins. Mainly, antibiotics have 5 different targets. They target the phospholipid bilayer and disrupt its function by increasing the permeability of the membrane, which affects the osmotic pressure of the cell and causes leakage of important biological molecules. Most antibiotics target the cell wall of the bacterium, and they inhibit cell wall synthesis. An example of these antibiotics are penicillins and cephalosporins. They also target DNA and inhibit DNA-RNA synthesis. An example of these includes quinolines. They also inhibit DNA-RNA precursor synthesis like sulfa drugs. They target ribosomes and inhibit the cell's protein synthesis. An example of these antibiotics include tetracyclines, macrolides, and aminoglycosides. Naturally, a bacterium resists an antibiotic by disrupting said antibiotic's mechanisms of action. This resistance is encoded within a bacterium's genes. Specifically, this resistant gene is located within the plasmid of the bacterium. The 4 main mechanisms of resistance are the usage of efflux protein pumps to eject the antibiotics from inside the cell, the synthesis of bacterial enzymes that modify the target of the antibiotic, the synthesis of bacterial enzymes that modify the antibiotics themselves, and the mutation of outer membrane proteins that allow the entry of the molecules into the cell. This will affect the permeability of the cell and prevent the antibiotics from entering the cell. So now the main question is, how do these bacteria acquire the resistant genes required to dismantle the antibiotics mechanism? Due to the vast numbers of bacteria in existence, a small percentage of them will have the acquired resistant gene by natural random genetic mutations that occur during bacterial reproduction and replication. The danger, however, lies in the bacteria's ability to transfer this resistant gene to neighboring non-resistant bacteria. These genes transfer by both vertical gene transfer and horizontal gene transfer. Vertical gene transfer occurs when the resistant gene is transferred to the next generation. Horizontal gene transfer occurs when the resistant gene is transferred across different bacterium genera. Due to natural selection, bacteria with this resistant gene will be favored at the expense of others because they are more likely to survive and reproduce and replicate. Eventually, most of the bacteria will be resistant to a specific antibiotic, rendering it useless. Humanity's method of counteracting these mutations is by introducing another chemical which disrupts the bacterium's method of resistance, so that the antibiotic delivers its functionality undisturbed. An example of this would be the addition of clavulanic acid to penicillin. However, the problem lies in whether our intelligence is capable of matching the bacteria's speed of mutation. This problem is further exacerbated by the misuse and overuse of antibiotics by both patients and professionals. At the end of the day, it's a battle between bacterial evolution and human ingenuity and intelligence. Done by Hamad AlShatti, Abdulrahman AlShuaib, Salman AlMutairi, and Abdullah AlKandari.